Professor Linrong. Professor Linrong Lu is, got his bachelor degree at the University of Science and Technology of China and PhD degree in Shanghai Institute of Biochemistry. He has his postdoc training in the Harvard MIT Division of Health Science and Technology in MIT, and be a research scientist at Dana-Farber's Cancer Institute, Harvard Medical Schools. In 2008, he is a professor of Institute of Immunology at Zhejiang University School of Medicine. And in 2015, he is the Vice Dean in Zhejiang University, University of the Edinburgh Institute. Professor Lu's research is to elucidate the molecular pathways and the cellular interactions that mediate and regulate stem cell development and T-cell mediated immune response. His work led to the discovery and functional characterization of two key signal molecules, TASPA1 and MINK1 kinase, which play a very important role in the stem cell development and peripheral T-cell effect functions. Professor Lu has published more than 60 academic research and review papers, which including Nature Immunology, Immunity, JAX-MI, PNS, and blood. So today, he will talk about the MINK1 kinase missing link between the environmental stress and the T-cell immunity. Okay, Professor Lu, you can begin.
Okay, thank you, Xing, for your introduction, and also thank Xiaolei and Chen for organizing this Immune Zoom seminar series. My topic today will be talking about a molecule we have been working in my lab actually for a long time, which is called MINK1 kinase. And my title will be Missing Link Between Environmental Stress and T-cell Immunity. I want to introduce a line of research we have been carried on to this molecule. And as you can see here at the bottom, a beautiful campus I spent 2 or 3 days every week. So I would want to have an advertisement first. We have built, actually collaborating with the University of Edinburgh in UK, we have built a joint institute aimed to raise biomedical students with both undergraduate and graduate program. And currently we have 2 major programs. One is biomedical science, the other is medical informatics. And we also aim to build a research team which could perform state-of-art biomedical research. So it has been, since its establishment, it has been 5 years now. We have built a pretty much international team. You can see the pictures of our PIs at the bottom, and we have built our co-facility and also our animal house, and we are actively recruiting. So if you have any postdocs who are looking for a job, you can consider this institute. And also the students we have are pretty good. So if you're recruiting PhD students, then we will provide a very good student resource. Okay, back to my research. As Xing has introduced, we have been working on T cells, interested in both T cell development, differentiation, and also how the effector functions are regulated, mostly in the context of autoimmune disease. One of the reasons is that we really have no condition to perform infectious experiments. So in the now 12 or 13 years since I established my lab, my work mainly focused on site selection and Th17 differentiation. And more recently, some work about tumor immunity. So I will first introduce the story of TESPO1 very briefly. It's not the major topic today, but I think it's a chance that I can sell my point. So since I was a postdoc in Dana-Farber, I characterized some which is called ESTs, expression sequence tags. This is something what we have before the Human Genome Project, basically the small transcripts. And by analyzing these expression sequence tags, I found a dozen of genes which potentially express in the lymph node organs, especially in the lymph node, spleen, and thymus. And I named them as TSEs, thymocyte-specific expression tags. So you can see a list over here. And after I came back to establish my own lab, most of these candidates actually has been studied by other labs. So we decided to study one of the candidates, which is the TSE3, the top 3 candidate on our list. You can see it expressed in lymph node, spleen, and thymus with very high expression level. When we make the knockout, we actually found that this mice has a defect in single positive cell development. You can see that the TSE3 knockout has roughly 1/3 of the single positive CD8 cells and CD4 cells when compared to the wild type, indicating a blockade in positive selection. And we later able to show that this molecule actually affect the calcium flux signal and the downstream NFAT activation. You can see the knockouts, after TCR activation, the calcium flux level is much lower than the wild type. And we also did some mechanism study to show that TESPO1 on one side is able to bind to the T cell receptor complex. On the other side, interestingly, it binds to a family of receptors on the ER membrane, which is the IP3 receptor family. Also the calcium channel in the ER membrane. So it explains why it affects the calcium flux from the ER to the cytosol upon TCR activation. And later on, we were able to understand how it works. We can, from the left, you can clearly see that the IP3 receptors are located on the ER. So in the resting T cell, it stays in a paranuclear region in the cell. However, upon CD3 engagement or TCR activation, here we use the anti-CD3 antibody on the glass slide to activate the T cell. You can see almost all the IP3 receptors goes to the position where the TCR is. So its function is to relocalize the IP3 receptor to the TCR complex upon TCR activation. When we knock out the TESP1 or make a mutation which cannot engage this interaction, you can see this translocation of the IP3 receptor to the TCR receptor is greatly reduced. So that's why, if we mutated this protein, we have the same phenomena like the knockout, that the calcium flux is reduced. So by doing that, we propose a model how TESP1 work downstream of TCR, which is that the TCR bind PLC gamma 1. I didn't show you the result, but it's upstream binding partner, and downstream it binds to the IP3 receptor on ER. So then it is very interesting or very useful, because now it brings the ligand where the IP3 receptor is a ligand for this calcium channel, which is produced by PLC gamma. And the IP3 receptor can accept this ligand at a place where it is generated. So it could happen very fast and very efficient. However, when we knock out TESP1, this recruitment or translocation of the IP3 receptor would not be able to happen. Then when the IP3 is generated, they need to diffuse to the IP3 receptor on the ER all over the place, then to trigger the activation of this receptor. Obviously, it will be much slower and less efficient. Okay. And more recently, what we found is that TESP1 expressing much higher level in the thymocyte, but much lower level in the peripheral T cells. And when we make a conditional knockout only in mature T cells, we find that the calcium flux in the mature T cell is not affected by TESP1 knockout. And the TCR signaling as well as TCR triggered proliferation of these cells were also not affected. And this is a negative data, but this negative data is actually very important for us to address the importance of the regulation mediated by TESP1. What we think is that we were able to answer a very important question in the TCR signaling area during thymocyte development. The question is, how do double positive thymocytes coordinate low PCR expression and low affinity cross-reactive ligands however, to succeed efficient positive selection? As you can see that a developing DP cells, when they undergo positive selection, their TCR expression level is almost 20-fold less than a mature T cell. And also the affinity which allows positive selection, the affinity is much lower than the antigens which can stimulate a peripheral T cell activation. So this means that the thymocytes need to be more efficient in TCR signaling. And what we found here, we think that TESP1 allows the sensitizing of this signaling process to make the DP cells more potent in TCR signaling, and thus allow the DP cells to fulfill the need of positive selection and these two circumstances, low receptor expression and low affinity ligand. So another significance we believe is that I was invited to write a commentary recently, and by analyzing the currently literature, I found that there are also some other molecules, not only the TSP1 we found, but also BANK in B cells and SLAP2 in T cells, which could possibly play a similar way. So we think that mobilizing ER IP3 receptor may serve as a general mechanism to enhance calcium signal, but we just don't know when it is needed and when it will happen. Okay, I just don't want to give up the chance to describe what I think is more, which is the most important finding I have since I established my lab, but I will go back to today's topic. The first part is the suppression of Th17 cell differentiation by MINK1, which is mainly carried out by a PhD student Guotong Fu and also student Yuan Junqiu and Qin Xu. Guotong's name might be familiar to you because he later on, after graduate, joined the Hongbo's lab to do postdoc, and his Nature paper is online this morning. So I would like to take this chance to congratulate both Guotong and Hongbo for the paper, for their great success. Okay, back to the Th17 story. it is well known that naive T cell, when they get activated, if there's a TGF beta and IL-6 in the microenvironment, it will drive the differentiation of Th17 cells. However, Th17 cells is also well known that they are often affected by environmental signals, including Wu work in 2013 to indicate that salt is one of the thing that can affect Th17, and also lipid and redox situation will also regulate Th17 differentiation. We accidentally go to this field because we were studying this molecule, which is a kinase, belongs to MSN kinase family, which is a small subfamily including NIC, TNIC, and MINK. We were interested in study it because it is, first of all, it is a kinase first characterized in yeast and considered as a MAP kinase upstream kinase. So it has another name, which is called MAP4K6. It is a MAP kinase. And there isn't much work done to this molecule. And in 2005, two major papers described its function. One is from a tumor lab, which consider it as a downstream kinase of reactive oxygen species in RAS transformed cell, and it will, through a complicated pathway, induce cell senescence. The other work is actually from the lab I was doing postdoc. Another postdoc in a lab showed that MINK can be activated on the TCR stimulation and mediate the negative selection of thymocytes. And this kinase is expressed both in CD4 T cells and CD8 cells, and it seems that the expression level in CD4 T cells is a little bit higher. And this is the data which is published by this another postdoc, and he, by using RNA knockdown, she was able to show that this is also using the HY model. You know, HY T cells usually are deleted in the matching MHC background male mice, but if MINK1 is knocked down, the cells are not deleted. That's why she claimed that it is a molecule related to negative selection. However, when I developed the knockout mice, I don't see any phenotype in the thymocytes. The thymocytes are completely normal. You can see here the cell number and the ratios are not changed when the MINK1 is knockout. More importantly, when I cross these mice to HY T cell receptor, you don't see a rescue of the DP positive cells, which is so clear that in the previous work. So that's why, you know, these mice, we couldn't do anything more on the thymocyte development part, and we start to analyze its possible function in the peripheral. So the first sign we observed is that in MINK knockout mice, there's a more CD44 high, CD62 low cells, this population, which is considered the effector memory cells, which is consistently high in the knockout mice. On the other side, the naive cell ratio is reduced in the mice, which indicate improper activation of CD4 T cells in the mice. If that happens, we of course will go ahead and check what happened. When we stain the cells with IL-17A and interferon gamma, and we found that in the cell, in the MINK knockout mice, the CD4 T cells are producing more interleukin 17, as well as more interferon gamma. And at the same time, the FOXP3 positive CD4 T cell number and ratio didn't change. We also tested the Treg activity in vitro suppression assay, and there's no change found. So when we go ahead, my screen is frozen. Oh. When we go ahead to check the lamina propria, we also find a difference of our 17 producing cells, that the knockout cells are producing more Th17 cells, while at this time we not only didn't see a difference in Tregs, interferon gamma generating producing cells also there's no change. So which indicate that the primary difference probably is due to Th17 differentiation. So we also use the in vitro differentiation assay to test their ability in vitro polarization, and you can see that no matter how much TGF-beta we use in different conditions, you can always see more Th17 cells in the knockout group, but much less in the wild type. And more interestingly, even without TGF-beta, you can already see around 10% of Th17 differentiation. This is quite similar to what Yisong has observed in their SMAD4 knockout. So we also see there's no difference in the vitro polarization of Th1, Th2, and inducible Treg. So then we want to know whether the increase the Th17 cells in vivo would cause the animal to be more susceptible to autoimmune disease. Of course, we use the EAE, which we are most familiar with. So you can see that in this diseased mice, you can always see more IL-17 cells in the central nervous system, and also the MINK knockout mice developed more severe EAE disease, and there's also more loss of myelin and also increased infiltration of lymphocytes in the inflammation site. And why MINK1 can regulate Th17? So we get some hint from a published paper, PNAS paper in 2011. A work which has been done in Drosophila has shown that MSN kinase could phosphorylate MAD. So MS in mammals is the MINK1, one of the family members, MINK1, and MAD is in mammal is the SMAD protein. So in all three, in the SMAD family, in mice, the SMAD2 has the conserved site, which has been shown to be able to regulate its activation. So we suspect that similar mechanism may also work in CD4 T cells. So the next few results just to prove that, we see that in MINK knockout mice, you do see more SMAD2 C-terminal phosphorylation or its activation upon TGF-beta induction. When, however, the T-threonine 324 phosphorylation is reduced in the knockout. Here, SMAD3 is a good control because SMAD3 don't have such residue, so its activation is not affected by MINK knockout. And on the contrary, if we overexpress MINK and SMAD2, you can see the increased phosphorylation in this specific threonine site, while you see decreased C-terminal activation. And if we mimic this threonine to a phosphorylated site, then the C-terminal activation was also blocked. We also use in vitro kinase assay to show that MINK, recombined MINK can directly phosphorylate SMAD2, but not SMAD3. So now we know that MINK can, through the phosphorylation of this threonine site, to block the C-terminal, which is exactly similar mechanism used in Drosophila. And this is to limit the inflammatory response. So we next asked what is the upstream regulatory function of MINK. So we take advantage of that molecular cell paper, which show that reactive oxygen species serve as an activator for this kinase. So we do think this matches to our observation, because in that paper, ROS has been shown to be able to activate MINK. And in previous research, ROS has been shown to be able to suppress Th17. And what we observe is that MINK could suppress Th17. So it is possible that MINK just act as a bridge to mediate this suppression. So by using ROS scavenger and acetylcysteine, we were able to show that if we use NAC, we add NAC in vitro polarization, you can see it can increase the Th17 differentiation to a level which is very similar to the knockout. While if the MINK is knockout, if you add NAC, then it will not be able to further promote Th17 differentiation. So what we consider ROS is actually the upstream regulator. So we can also achieve that by feeding the mice with NAC in the drinking water for 2 months. And when after that, if you check the Th17 cell ratio in the lamina propria, you can see increased Th17 cells to a level very similar to the knockout. Also, again, the knockout didn't show this obvious increase. And also, if we purify the CD4 T cells from this NSC-fed mice, they were able to mediate or generate more severe disease than the wild type, but which is similar to the knockout cells. So by this point, there's another question we actually cannot answer, which is how ROS could activate MINK1. There's no examples that a kinase could be regulated in a redox condition. So one of the major targets for ROS is actually phosphatase. So this led us to be interested in studying the role of PP2A in Th17 differentiation. It has been, what I haven't shown you is that the SMAD2 threonine 324 site actually could be enhanced by a chemical which is called OA, which is the inhibit of PP2A. And in another biochemical study, which has shown that PP2A could coexist with MSN family kinase in a big signal complex called STRAPAC complex. So it is possible that ROS is not directly activate MINK, but it is through the inhibition of PP2A. So that's why we went on to study the role of PP2A in Th17. And we were able to knock out the PP2A catalytic domain, the PPP2C alpha in the T cells by using a LCK distal query. And after that, if we do a T cell polarization, you can see that PP2A knockout cause a decrease of Th17 cell differentiation, but do not affect the other types, including Th1, Th2, and Treg, as what we expected. And PP2A expression level has been associated with the disease severity of SLE, as well as some expression profile of Th17 related molecules. So we also were able to show that PP2A knockouts in T cells will cause less severe EE disease, and as well as we can use PP2A inhibitors. We tested different inhibitors of PP2A and found one of them, the name I don't know, I cannot remember the English name. The Chinese name is Banmao Shu, which is very effective in treating this disease. So as a summary for this first part, we have identified MINK as well as PP2A as a regulator of SMAD2 activation, and thus able to regulate Th17 mediated inflammatory disease. So my second part, I will briefly introduce its role in NAP3 inflammasome activation. The reason why we carry out this study is that after we published the MINK deficient mice phenotype, we are frequently asked whether this knockout mice led to autoimmune disease, spontaneous autoimmune disease, but we didn't find it. And one of the reasons might be that MINK1 is expressed in other immune cells, so there's other functions, and that's why we see that the knockout is overall even healthier than other mice. So this work is carried out by another PhD student, Kai Xiang, with the help from Xuexiao. And what we able to see that the MINK knockout do lead to less NAP3 activation. You can see the R1 beta production as well as caspase-1 activation were reduced in the MINK knockout mice, as well as the production or secretion of R1 beta and R17 were also reduced. And the MINK knockout mice is also more resistant to sepsis induction, and there's more mice be able to survive in sepsis. And you can see the R1 beta and R18 level in the serum is also lower, although TNF alpha and R6 level is similar. So we were able to characterize, because MINK is a kinase, so we were able to show that there's a phosphorylation site which is directly mediated by MINK, which is the serine 725 site in mouse NAP3, which is equivalent to the 728 site in human NAP3 molecule. And by doing this research, we were able to conclude another function of MINK in macrophage, which is induce, I think I have a few slides missing, but we also proved that MINK1 is also downstream of our acid macrophage to basically prime the NAP3 activation through this specific phosphorylation site. We think that our model, our research provide a very precise mechanism for RS, how RS could activate NAP3, which has some, hypothesis, but it's kind of complicated to us. Okay, so I'll go to my last part. I will briefly go through it. Dr. Kai Xiang want to challenge mice more, even without my notice at the beginning, to challenge the mice with some graft tumors to see if the knockout will affect the anti-tumor response. And luckily, what he found that is that if you engraft EF4, EG7, which is the EF4 expressed over, if he grafted these tumors into the white type and knockout mice, you can see the knockout mice grow smaller tumor, which is which is actually a consistent result. You can see less tumor volume, indicating enhanced anti-tumor immunity in these mice. And similar results were obtained using B16F10 mice through vein injection. You can also see better survival of the mice. Also, if you sack the mice earlier, you can see less tumor module in the lung. And when Kaishan started to analyze what happened inside the tumor, he was able to see that the MINK knockout leads to the appearance of a specific family of population of CD8 T cells, which is absent in the MINK wild type mice. So it indicates that CD8 function difference is involved in the enhanced anti-tumor immunity in the knockout mice. So this is just correlation analysis to show that the increased number of CD8 T cells in the MINK knockout mice correlate well with the reduced size of the tumor. And the next two experiments is to use the experiment to actually prove that it's CD8 cell, which is work. The first experiment, of course, is the CD8 depletion assay by giving multiple doses of anti-CD8 antibody, then to challenge the mice with EG7, Kaishan was able to show that after depleting the CD8 cells by the antibody, you can see that both knockout and white mice, in both knockout and white type mice, the tumor grows much faster, but more importantly, the difference between the white type and knockout mouse disappear. And another experiment which were used is to use the adaptive transfer mice, which is also widely used. Kaishan purified CD8 T cells from the wild type and MINK knockout mice, transferred them to the reg knockout host, then challenged the mice with the tumor. And you can see the mice receive wild type CD8s, the tumor grows bigger than the mice receive the MINK knockout CD8. So this is very similar to what we observed in the straight knockout mice. By analyzing the CD8 cells, we were able to show that MINK knockout CD8 cells are better effector cells. They produce more TNF-alpha, interferon gamma might be a little bit more, and also more granzyme. And if we mix the wild type and MINK knockout cells in a 1-to-1 ratio and put them back into the rag mice, and you can see that the MINK deficient CD8 cells has the advantage in expanding in the host. But something strange is that, not only in the tumor, it seems that in the lymph node and the spleen, the knockout mice cells even have more advantage. Of course, the cells with receive knockout CD8 plus the CD45 control mice, the tumor also grows smaller than the one receive both wild type and wild type CD45 mixed mice. So we will also be able to show that the CD8 T cells have enhanced the killing capacity in two different assays. One is a killing assay against LCMV infected candidate target cells, and another assay where OT1 cells were primed in the same host and used and isolated to used to kill I think it's the EL4 targets. In both cases, the knockout cells have higher killing capacity. And I have mentioned to you that we have found this population of CD8 cells which appear in the knockout, and it turned out these are CD8 cells showed less exhaustion status. They have less expression of most of the inhibitory receptors. When we check with FACS, TIM3 is the most obvious one. You can see very substantial decrease of TIM3 expression to a less extent, but it's also true for PD1, for TIGIT, and LAG3. They are less obvious, but they are all reduced. So if we check the expression pattern of these CD8 cells, we find, if we use GSEA to analyze the PD1 blockade effect signature, you can see that the gene expression pattern of this PD1 blockade is also could be found in MINK knockout cells, which means that MINK knockout can reach a state of less exhaustion. So then we want to compare, we want to see whether anti-PD1 therapy will, how it will, work. So we use the anti-PD1 antibody in both wild type and knockout mice. You can see the antibody could increase the cell infiltration to the tumor, but what we found is that the anti-PD1 antibody treatment can only reduce tumor size in the knockout mice, but not in the wild type mice, but couldn't further reduce the tumor size in the knockout. And this result might look disappointing, but I think it's reasonable to what we have found, because the MINK knockout mice are already less exhausted. So that's why, it couldn't be further reversed by the PD1 antibody. So we are still, this is unpublished data, we are still trying to working out the direct target of MINK1, but currently we are exploring its role in T cell senescence, because when we analyze the RNA-seq data, we can see that the senescence signature is genes are enriched in MINK knockout mice, including oncogene-induced senescence, oxidative stress-induced senescence, and also DNA damage-induced senescence. Basically, its senescence is reduced. So senescence is a state which is similar to exhaustion, but it's not totally similar to exhaustion. So we did some analysis to show that in CD8 T cells during aging, there are actually less CD44 high cells, which is considered as a sign of aging in mice. And when we analyze the CD8 inside the tumor by using this CD27 negative, CD28 negative, and KORG1 positive as a marker, you can see there's less cells, less senescence T cells in the knockout compared to the wild type. And of course, we see less gene expression level of P21 and P16. And we also consistently see that this senescence reduction in different experiments, and we also managed to stain the cells with beta-galactosidase activity and show that MINK knockout mice has less staining, which also indicate less cell senescence. And this senescence has been shown to be able to be induced in vitro by co-culture CD8 pre-activated CD cells with tumor cell supernatant. By doing that, we can also see less induction of this senescence the cell in the knockout. You can see the level is always lower than what induced in the wild type. And this also results better proliferation capacity after co-culture with this supernatant, which is a function readout of less senescence. So this is so far what we have got in our experiment, which is still ongoing. So by summarizing all three parts, we are now suspecting that MINK kinase may serve as a downstream regulator of ROS, and thus to play by working on different substrates to play cell type specific roles in mediating different cellular response. So this is all I want to share with you today. So I already acknowledged the students who performed these experiments, so I can stop and answer questions. Thank you. Okay, so I finished.
Okay, thank you for Professor Lu's very interesting talk. So if anyone has questions, you can type your questions in the chat so I can ask one by one.
Hey, Linrong, this is Xiaolei. This is really a beautiful talk. And also I learned a lot. So I think I have a few questions. So for your first part, the relocalization of the IP3 receptor to the immunological synapse in the thymocyte. So did you also see a similar like relocalization in these like peripheral T cells of the IP3 receptor?
Okay, so can you hear me?
Yes.
Okay, and yes, we can see similar, translocation of the IP3, but it's much, it's weaker. So, for example, we did the experiment in Jurkat cell. We can still see it, but when we overexpress TESP1 in Jurkat cell, we can see much stronger translocation, but the translocation will be very weak if we don't express TESP1 in Jurkat cells.
I see. So is TESP1 also like normally expressed in peripheral, let's say CD8 or CD4 T cells, or its expression level is lower as compared to the thymocyte?
Yeah, it is true that the highest expression is in DP cells. Okay. When it mature into single positive cells and when they go to the peripheral, the expression is still there. You can still detect it with Western, but when we compare to thymocyte, it's much lower, like at least tenfold.
I see.
Yeah.
Do you know where the TESP1 like intact with PLC gamma? To still remember that, or it had been mapped out?
Yeah, we mapped out. What can I see? Yeah, we cannot map the domain, but we map out a little motif called PFF motif in the TESP1. So there's a if we mutate these two residue, then the binding is gone.
I see. Do you know like which part on PLC gamma one that bind the
PLC gamma? It's interesting that we also mapped it, and it is the catalytic domain. Oh, so what we think domain. Is that because after T cell activation, the TESP1 was recruited into TCR. So we think only this happened only after the conformation change of the PLC gamma 1 when the catalytic domain is exposed.
I see. Okay, that's actually very interesting. Because for PLC gamma 1, so one of its special is it has this split like a catalytic domain, which is like spaced by these SH3, SH2 domains. Do you think like the TASPER1 will bind to other PLC family of protein in addition to PLC gamma, like PLC beta, or because
we don't know. We didn't test this in other cells, but it's possible, you know, if it's expressed, yeah.
Okay, that's very cool. So my other question is about the MINK1. So is there any like consensus motif for its substrate?
Right now, we don't know. You know, the TGF beta, the SMAD2 phosphate site was originally identified by other labs. We just copied that, but we made that phosphor specific antibody, and were able to see that. And for NAP3, we identified by mass spec. But you know, it's also a overexpression, co-expression condition. So we don't know whether it's also phosphor, you know, other substrate. And in the CD8, we currently don't know. We want to do a phosphor analysis to see whether we can find its substrate or not. But whether they have a, you know, similar pattern or structure, we don't know yet. We are not at that stage yet.
I see. So is the structure available?
Not really.
Not really, okay. Also, I just wonder if there's like, you can make some catalytic dead mutant then serve as a trap to like pull down these like substrate. If the structure is available, then that can be designed properly.
Yeah, that might be an idea, but we are currently going to another condition because right now we consider it might be a kinase which was targeted to. So we want to select some small molecule drugs to see whether we can find one to, you know, mimic.
I see. So that's very cool. So is MINK1 like homogeneously expressed in the cytoplasm, or it has specific location?
You know, it's we actually hasn't looked at very carefully. We just you know, according to, you know, the published data, it seems to be, we don't see a specific pattern for it. It's more like ubiquitously located. Yeah.
I see. So the phosphatase that antagonize the MINK1 phosphorylation. So is that the PP2A or other phosphatase?
Yeah, we think it is PP2A because of the biochemical evidence provided by other groups, as well as our function, you know, its opposing function in Th17.
Okay, that's great. Okay, cool. Yeah, thanks so much, Linrong. This is a really beautiful talk.
Thank you.
Okay, so Dr. Lu, Jeff Zhu has questions. His question is, do this MINK knockout anti-tumor CD8 T cells express IL-17 or display stem cell-like phenotype? This cells also has connection to the TGF beta signaling.
Okay, yeah, I saw Jeff's question on the notebook, but to all these questions, I don't have answer. We haven't checked whether the CD8s can also express IL-17, and you know, I know this stem-like or exhaust stem-like cell description, but we haven't looked at that. And right now, we don't know whether it will connect to TGF beta signaling or not. You know, Jeff, if you have more detailed in suggestions, probably you can tell me, you know, if not today, maybe another chance. I do need input in this project.
Yeah, it's a great talk, actually. You know, the reason I'm asking just because CD4, CD8 often share some similarities. Since you observe increased TS17 response, and so it's easy to imagine that whether there's some IL-17 production, because also IL-17 may also be involved in anti-tumor response, and some stem-like signaling may be also related to, there are reports that if you were to culture or in under TS17 like condition for CD8s, and those cells actually grow much better and have much higher anti-tumor activity, even though many of them will eventually switch to interferon gamma producing cells. But the initial step of differentiating towards IL-17 production phenotype may allow them to stay, like serve as a reservoir of the effect of CD8. So it might be.
Okay. Yeah, it's not hard to test. We should test that. Yeah.
Good. Thank you.
Thank you. Yeah.
Okay, so the question from the Jeff Young is, does MINK knockout affect other SMAD besides SMAD2?
Okay, so for this question, because we only interested in the TGF beta signaling during Th17 differentiation. So what we have tested is, of course, it's SMAD2 and 3, among which SMAD2 has this throning site, so it is regulated by MINK1, while SMAD3, as I mentioned, they don't have this throning. It is alanine in this site, so it is not affected by MINK1. So I don't know, is that enough?
Okay, so does anyone have more questions?
Hey, Linrong.
Hey, hi.
I have questions related to the MINK activation. You say it's actually upstream activated by ROS, right?
Yes.
Yeah. So my question is, why Th17 specific? Because as far as TCR activated, the ROS will potentially activate the downstream signaling, and then this is not only specific to Th17 cells. So the other T cell subset also get involved with the ROS activation. So why MINK only influence the Th17 cells?
Okay, this is a tough question. I just tell you what we think. Okay.
Yeah, sure.
So I think one thing is that we don't say it is Th17 specific because in this context, it's, by working or by targeting SMAD2, it will affect, the cell event which is related to SMAD2. So it turned out to affect SMAD2, and SMAD2 is involved in Th17 differentiation in this condition. So this is why in the first story we find, it eventually target to Th17 differentiation. And another thing I want to mention is that ROS is a very complicated molecule. It affects so many things, and the concentration of the ROS is really the key. If there's no ROS, the T cell probably wouldn't be able to get activated. If there's too much ROS, it is also very well known that it will hurt any kind of cell. The cells won't be healthy and they just won't work. And what we have proposed in that story is that we are not dealing with these extreme conditions. The condition we consider where it can modulate SMAD2 and Th17 is the physiological level of ROS, which is not posing a stress or a harmful stress to the cell yet. Because when we culture for example, when we culture the cell, when we polarizing the Th17 cells, then titrating different concentration of ROS. During the concentration we use, I think it's up to 5 millimolar. It enhanced Th17 differentiation. However, when we increase the concentration, if it's more than 10 millimolar, then it starts to inhibit cell growth as well as Th17 differentiation. So the concentration here is, I think, somehow very important, and that's why we consider there's a window. This is the window where MINK1 might sense or get activated and playing a role in this window of ROS concentration. So I don't know if my answer satisfy you, but this is what we think.
So if that's the case, I have following questions. So first of all, do you see MINK activated in other T cell polarized condition besides Th17?
I would say yes. MINK activation, whenever we activate the T cell with TCR, we always see MINK get activated.
Okay. Then on the other hand, the transcription of MINK, is there any change in different T cell subset?
I think overall there isn't big difference between the expression level. There's some variations, but there's not dramatic.
So all the downstream effect is due to the phosphorylation.
Yes.
And then because so if I understand this correctly, you believe that the reason the specificity of MINK on the Th17 cells is just because of the phosphorylate target of the MINK. Different in them. Okay. Okay. Thank you. Yeah.
Put another way to answer your question, because MINK activation is somehow regulated by PP2A, so it will make the story more complicated because PP2A regulates so many different substrate, almost like 1/3 of the cellular phosphorylation. So that's why MINK1, if it's bound to it, it might, if it's with different regulatory subunit of PP2A, it might be able to preferentially, work on different substrate in different cell type.
Okay. Okay. Thank you.
Thank you.
Okay, so if no one has more questions, thanks again for Professor Linrong Lu's talk. And today's seminar will stop here. And thank you all for your listening. Thank you very much.
Okay, thank you, Zhejiang.
Mm-hmm.
Yeah, nice to see you all.
Thank you.
Yeah.
Yeah.